Okay, we'll go ahead and get started. So good morning, everybody, and welcome to Heart Day. Very happy to see a good crowd here, and it promises to be a really enjoyable and insightful day, I'm sure. So we're going to go ahead and get started now, and it's my distinct pleasure to introduce our morning speaker, Dr. Katherine Yutzey. Dr. Yutzey, she did her PhD from Purdue in 1992. And since then, she has been a professor at the Cincinnati Children's Hospital Medical Center. Her research area primarily is in cardiomyocyte proliferation, and she's very, very well known in heart valve disease, specifically looking at molecular mechanisms of various pathologies in heart valve disease. And I dare say that I think in the field of heart valve disease, she's probably, I would say, the top 5 most well-known folks, I would say. I first met her when I was at Pittsburgh, 2007, I believe. And so she continues to also inspire a lot of younger scientists, and she's definitely been a mentor and inspiration to a lot of younger scientists. And she does also involve herself a lot in the mentorship aspect of things as well. She's fellow of the American Heart Association, fellow of the American Association of Anatomists, right? And she has several other accolades, including, of course, being extremely eminent scientist in the field. So without further ado, please join me in welcoming Dr. Katherine Yutzey.
Okay. Oh, this works. All right, so thank you, Sharon. And all right, where's this one? Okay. Where do you need it here?
Yeah, just away from the speaker.
All right. All right.
Yeah.
Is this gonna work? Okay. So thanks to Sharon and Josh for the invitation. I'm very honored to be invited as your keynote address today. And what I'll tell you about today are some of our recent work on heart valve development and disease mechanisms. So just to go back and have a good foundation for heart valve development in the embryo. I think this is not. No, it's not going to work. Okay, so in the embryo, the heart valves. Oh, this is. All right, so I'm going to just point. So the heart valves initially start out as endocardial cushions that form in the. I could probably just go. So these start out as mesenchymal structures that form in the AV canal and the outflow tract through largely an endothelial mesenchymal transition that give rise to these mesenchymal cells, but there are also valve cells that come from other origins, such as villal crest and epicardial cells. And I'm not going to get really into that, but talk more about what happens after the cushions, where they elongate as mesenchymal cells, and then they become stratified into layers. And so those layers are formed of collagen on the fibrosis side, which gives the valve sort of strength, proteoglycans in the middle, which gives the valve sort of sponginess, and then elastin on the side of flow that gives the valves elasticity. And so that organization of the valve is really integral to its function, and it goes bad with disease. And so adult valve disease is often calcification of that forms nodules that leads to stenosis, and it impairs the valve to be open and closed. And then what we've focused on more recently is myxomatous valve disease, where collagen and proteoglycan, those layers, become less distinct and they kind of merge together. The valve becomes thickened and can often become regurgitant or insufficient because it's lost a lot of its strength. And so congenital defects can lead to both of these conditions. Genetically, myxomatous valve disease has been linked to Marfan syndrome, which I'll talk about today, and also filament A and cilia pathways and extracellular matrix proteins. So these can all contribute to the degeneration of the valve over time, and often leading to adult valve disease, which someone may have a mutation in one of these genes and they don't really see it until much later in life when that valve needs to be replaced. And similarly, pathologic calcification is generally considered to be sort of a disease of the old or the aged. But bicuspid valves, which is a congenital malformation of the valve leaflets, can also predispose the valve to calcification. And so this congenital defect may show up in somebody who's 50 or 60 years old, again, who needs to have their valve replaced. Okay, so I come at this from a developmental perspective, and so thinking about how valves are made, we worked in chick on the top and mouse on the bottom, and these are embryonic days where you can see that these endocardial cushions, which start out as sort of blunt mesenchymal cells, elongate, and during that process they're dividing, so you get a lot of the development of these valve progenitors during this period of development, and then after that the cells sort of stop dividing and they start making matrix, and then you get this stratification that's later. And in the mouse, that happens actually after birth. And so here again, looking at these, you can see the cushions that elongate, but trying to see when the collagen really gets deposited in the valve and elastin, that's after birth. And so this is a mitral valve with the yellow stain from the trichrome. There's a lot of development again that happens after birth, and that may be subject to blood flow or hemodynamic forces or sort of the mechanical requirements of the valve during that period. And then this is work from Elena Akawa from several years ago, looking at human, and again, looking at the collagen organization that you can see on the bottom, sort of this fetal stage, early in development, there's really not very much collagen, but then the collagen undergoes a lot of remodeling and organization, and even from childhood to adulthood, you can see that you get a lot more layers in the adult valve than you get in a valve of a child. And so, again, this remodeling of valves is a process that happens during development of the heart after birth, especially in children and neonates. And one of the things that happens with disease, as I mentioned, is that the normal organization and the cellularity of the valve becomes disrupted. And so this is pediatric valve disease, an elastin mutation that's associated with Williams syndrome. And this is an aortic valve that is much thicker. If you notice the scale bars, it's a lot bigger. And the matrix has become disorganized, and there's this extracellularity. And so a lot of the overall organization in terms of the matrix and the cells are things that become disrupted in disease. So we're very interested in trying to understand the cells that are in the valve, and then also how these different cell types could contribute to disease. So one of the things that we did recently was to look at a single cell RNA-seq analysis. And so what we did was we looked at embryonic valves of mice after birth. So we grouped the aortic and the mitral valves together, and then also at P7, and so you can see they're starting to make those layers, but they're not done yet. And then at P30, where you do see these different domains of extracellular matrix in the valve. So we chose those two periods, and then we did single cell RNA-seq, where basically you dissociate the cells and you figure out all of the genes that are expressed in an individual cell. Well, not all, but overall in the population, you can get basically a picture of all the genes that are expressed in these different cell types. And so we used a drop-seq analysis. So Steve Potter, who's downstairs from us, helped us with this. Lexi Gould's a postdoc, and then Nathan Solomonis did a lot of the bioinformatics. And so basically what you do is you dissociate your tissue, and then through this drop-seq protocol, you add distinct barcodes on individual cells, and then you put an individual cell in a drop and try to get millions of reads out of these individual cells. And the idea is that you get a picture of what genes are expressed in these individual cells, and then informatically you can try and figure out what type of cell they are and what genes they're expressing. And so doing like a principal component analysis, sort of in an iterative way, looking at what these genes are and what we know about genes that are expressed in interstitial cells, so like type 1 collagen, immune cells, which was like a CXCR1 receptor for cytokines, and then PKM or CD31, which is a endothelial marker. We could group these cells into sort of these basic categories. And then for Lydia, we also looked at DCT melanocytes, and we found those cells as well in the single cell analysis. And they express legitimate melanocyte markers. And so here from the T-SNP plot in the left, you can get an idea of how many cells were in each population as well. And so you can see the interstitial cells were the majority of the cells, but then we got significant components of the populations as well. And looking at the percentages, so these are some of the genes that were sort of defined, these different cell types. And one of the things that really came out of this is that there are a lot of immune cells that live in the valves. And so about 10% of the cells that were in the valves are immune cells. And then about 10% of the cells are endothelial cells, and the rest of the cells are interstitial cells, identified really by expression of type 1 collagen and some of these other fibrous gene markers. So, and the percentages don't really change that much at the RNA level, comparing the P7 and the P30. So for the melanocytes, which was that smallest population, we saw that they do express some of these melanocyte markers. So DCT, tyrosinase, PMEL, MITF, and they are located where we had seen them before as dark cells if you analyze the valves of pigmented mice. You do see these dark cells, and so we always like, well, what is that? And then from the gene expression analysis, they do express solid melanocyte markers. And you can see that they are present within both the aortic and mitral valves, and they're distinct from the immune cells that we're marking with CD45. So it's a distinct population. Melanocytes makes melanocyte genes. We don't know what these cells are doing. Maybe Lydia will tell us that at some point. So I know that they've been working on
Sun out. Sun.
Okay, all right. I look forward to your poster. Yeah, so that's what we know about the melanocytes. One of the things that came out of this that was a little surprising was that endothelial cells are of distinct populations. And so we can see different types of endothelial cells based on their gene expression. And we were able to sort of group these into 3 different types. And we were calling them lymphatic, based on PROX1, which is a lymphatic endothelial marker. And then I'll show you why, but we also labeled one of them coaptation VECs, and then another VECs that were in different parts of the valve. So interestingly, these don't really change over time in the P7 to P30, pretty constant, but there are all these different populations. And where they're located in aortic valves, and we saw similar in mitral valves, is the what we were calling sort of the generic endothelial cells. They are located on both sides, but really near the belly of the valve. And then the coaptation ones, which are expressing some other extracellular matrix genes that may give them a little bit more resilience and maybe some Wnt signaling, was in the area of the valve where the valve's leaflets come together. And then the lymphatic endothelial cells are on the side away from laminar flow. And so we think that these endothelial populations may be defined based on the flow patterns or the fluid forces that are acting on these parts of the valves. And that's something that we're actively pursuing. But right now, we don't really know how these different populations maybe affect valve development or disease. That's something that we're working on actively right now. So looking at the interstitial cells, you can, in the valve, we know that there are different extracellular matrix genes that are, or proteins that are expressed in different parts of the valve. So we wanted to see, are these different cell types maybe that are producing these? And so at P7, we were able to see that the cells actually segregate separately from the P30 cells. So that means the P7 are in blue and the P30 are in red. And so that means that even in this overall cluster of interstitial cells, the P7 and the P30 cells are different, and they cluster together with one another, but they're not all intermingled among each other. So the P7 cells seem to segregate into two populations, one of which has more sort of glycosamine glycan kinds of genes in it, and the other one has more fibrotic or collagen related genes in it. And so what we're thinking is that even at P7, there are distinct populations of cells that are expressing these different extracellular matrix components. And then looking, this is looking at the sort of localized gene expression, and you can see that we see localized collagen versus versican, which is proteoglycan, at P7 in both the aortic valve and the mitral valve. And based on the numbers of cells, there are a little bit more of the GAG-producing cells than the collagen cells, but we do see increased numbers of genes in collagen versus glycosamine glycan cells. And so this gives an idea that these interstitial cells are probably doing different things in different populations. What we don't know is, are they sort of determined to do one thing, or is there plasticity in these cells? And that's something that we're looking at in our disease models to see if maybe when a valve gets thick or expresses more proteoglycans, then maybe this population changes. And so that's something that we're working on now. And then if you look at P30, when the valve is a little bit more mature, then we see different types of cells. So these cells are not the same as the ones that we see at P7. And they are expressing complement genes. We also saw an interesting population that is similar to scar-forming cells. So Jeff Mulcahy in our division has been looking at cells that are involved in scarring after myocardial infarction. And he's identified this population called matrofibrocyte that has sort of osteochondral kinds of genes like CHAD and fibromodulin, and this population shows up in the valve. And so I think that, again, it's a collagen-rich fibrotic structure, and so I think that's why that population is there. We also see a fibroblast-like population and a cell that has complement that may be involved in sort of immune surveillance. And so I think these are sort of the quiescent cells in a normal valve after it's undergone a lot of its remodeling. And then the other populations that we were very interested in were these immune cells. And so they do seem to change from P7 to P30. And at P30, the majority of the immune cells seem to be macrophages. But we do see distinct populations of these cells. But for the most part, they are more of the monocyte-derived cells, the macrophages, dendritic cells, and not so much T and B cells. And they are they represent about 10% of the cells in the valves, and they express a lot of these markers that are very characteristic of different immune cell lineages. So this intersected with another project that was going on in the lab, trying to understand really where are the immune cells in the valves, what are they doing, how are they localized, how do they get there? And so this is an aortic valve looking at CD45, which marks all immune cells. And so it looks like there are different localized populations, potentially on the area of the valves where they come together in that coaptation region, and then also on the other side of the distal tips. And so in our sort of speculation, we think maybe they're regulated by forces or hemodynamics. We don't know right now, but a question is, what are they doing and why are they there, essentially? So, and then the other big question, which I'll get into, is are these potential cells, if they are involved in disease progression, is anti-inflammatory therapy a potential way to prevent or to stop the progression of valve disease? So I'll get into that as well. So what are these cells? Based on our single cell, we knew that they were probably mostly of this monocyte origin, so monocytes, macrophages, dendritic cells. And when we did a lineage trace with CXCR1 Cre, which marks monocyte-derived cells, we found that most of the CD45 cells were in fact of this lineage. So again, not that many T and B cells. This is in a normal valve, but these cells are these monocyte-derived cells, and again, they're localized here, which is where the valve leaflets come together, and then here potentially where the valve, sort of the blood, kind of when the heart is in diastole, when the blood is kind of sucking back down on the valve, that is when that's on this side. But again, we're thinking this is related to hemodynamics, but we just don't know at this point. And then looking at these over time, so we looked at, again, P7 and P30, which is this postnatal maturation period, and then looked in adults and tried to figure out sort of what kind of macrophages they were. The ones that are sort of developmental are the ones that are more CD206 positive, and then over time, during this postnatal period, the bone marrow takes over, and in a lot of organs, the resident macrophages come from the bone marrow, and they are this MHC2 population that you see increasing at P30 to P60. So it looks like the macrophage origins and types of macrophages are changing during this postnatal period. And again, we don't really know what they're doing, but there's some changes that happen. So several years ago, somebody from in the South Carolina Visconti group had said that valve or sort of cells from the circulation go into the valve and they turn into interstitial cells that make collagen and extracellular matrix proteins. And so we were interested in seeing if that was the case in the cells that we were looking at. And so what Alexei Hulin, the postdoc, did was to take CD45 cells and sort those by FACS sorting away from endothelial or CD31 negative, CD45 negative. So they would be not endothelial cells, not immune cells. Compared those cells, which should be the interstitial cells, to the CD45 cells, which should be the immune cells. And what she found was that by RNA, the immune cells are expressing a lot of the chemokines and cytokines that you would expect from immune cells, from macrophages, and they do not express extracellular matrix genes, periostin, collagen, Versacan. And so what that said to us was that the CD45 cells that are present within the valve stay as immune cells, and then they do not convert into interstitial cells from this lineage trace. So, so the idea that these cells migrate to the valve and turn into interstitial cells, we think is not true based on the data that we have here from about the 1 month after birth. So based on the single cell and this immune study, what we know now is that there are distinct interstitial cell lineages and also distinct endothelial cell lineages. And I think from a tissue engineering standpoint, this might be bad news because you need to think about really the diversity of these populations when you're trying to make a construct that recapitulates in a normal valve. And then we're also very interested in what are these resident and infiltrating macrophage populations doing? Do they have a role in normal development and then also in disease? So we've been working a lot at this question with myxomatous valve disease. And the model, the disease that we've been looking at is related to Marfan syndrome. So this is a mutation in fibrilin-1, which is a connective tissue gene that can affect collagen structure. It's a genetic disorder, and the main manifestations of this are cardiovascular disease. In older individuals, aortic aneurysm is a problem that needs to be managed and monitored. But in younger individuals, mitral valve disease is a major problem, and in infants with Marfan syndrome, the mitral valve disease is the main thing that needs to be paid attention to in young infants. So right now they are managed using beta blockers, and there was a big study from, it was a big pediatric heart network study based on some work from Hal Dietz's group that suggested TGF-beta signaling was involved in this disease. And they did a big clinical trial with atenolol with losartan, which is a TGF-beta inhibitor, versus atenolol, which is the beta blocker therapy. And they didn't find any significant difference between these two treatment modalities. So we're sort of back to the drawing board in terms of what possible therapies might be used for these individuals. So the myxomatous mitral valve disease, so this is a normal mitral valve. You can see that it has leaflets and these chordae tendineae that basically keep the valve from turning inside out. And here again, you can see nice stratification and organization of the valves. And then in mitral valve disease, this is probably a Barr syndrome valve, the valve leaflets again get very thickened. They have a lot of proteoglycans in them. They're very squishy and sort of regurgitant. So these valves right now in older individuals are a major source of valve disease in around the world, essentially. So right now, the treatment for this is really surgical valve repair or replacement. And for mitral valves like this is a major open heart surgery that causes a lot of complications. And so this is a major clinical need, unmet clinical need, essentially. So we went back into mice models to try and understand what some of the molecular drivers of Marfan syndrome were. And we looked at Hal Dietz's original gene-targeted mouse model. This is before the day of CRISPR, so they had to do a homologous recombination. And these mice exhibit a lot of the skeletal malformations of Marfan syndrome, but also the aortic aneurysm. And this was the model that was used to look at the TGF-beta signaling in the aortic dilation aneurysm in these animals. So the valves in these animals are also highly myxomatous, and so this is comparing the wild type versus the fibrillin mutant. This is about 4 months in a mouse, and what you can see is that the mitral valves are thick. They have increased proteoglycan, and the collagen proteoglycan organization is disrupted. And so here, this is looking a little bit earlier. The green is the proteoglycan, the red is the collagen, and what you can really notice is that there is this mixing of the proteoglycan and the collagen, which is really sort of the structural basis of why these valves are insufficient. So what's driving this? In the field, there has been talk about endothelial mesenchymal transition as a driver for valve disease. And so we initially started this trying to see if some of the increased numbers of cells and the increased matrix that we're seeing was from a reactivation of endothelial mesenchymal transition. And so what we did was a fate mapping study where we labeled the endothelial cells at P0 with a tamoxifen-inducible CRE, and then looked 2 months later to see if any of those endothelial cells had migrated into the inside of the valve. And so what you can see is we don't see any of these interstitial cells that are expressing this green endothelial marker, even in these fat valves. And so what that said to us that really EMT was not a major driver. at least in this model at this time, with the endothelial fate mapping, which I think is really the definitive marker for an endothelial mesenchymal transition. So, at least from this, in this situation, we're confident that there is no EPT that's driving this disease. So what is it? So what we did was an RNA-seq analysis. So again, here we just took the whole tissue and then did RNA sequencing of the whole batch tissue, so all the cells are together. But what it did tell us was that there were increased markers of inflammation and increased markers of macrophages in the fibrillin mice versus the normal controls. And so that suggests to us, well, maybe there's an inflammatory component in the progression of this disease. So looking at the potentially infiltrating immune cells, again, looking at CD45, which is a good marker, basically all the immune cells, what we found was that there are increased in CD45 cells, the green ones, in the areas of the valve that are thickened. And then again, we did see our old friends, the CD45 cells that are normally expressed in the valve. So in the fibrillin valves, there are more CD45 cells and they're in the wrong place. And then trying to understand what types they are. So in the wild type, we see two different kinds of macrophages. So the CD206 cells are up here, the MHC2 cells are more down in the distal tip. And in the fibrillin mutant valves that are thickened, we see increased expression of both of these populations, and they're sort of commingled where they shouldn't be, essentially. So what that says is that not only are there increased numbers, but dysregulation of where they are, and there's a lot of mixing of these cells. So the macrophage population seem to be very dysregulated. We didn't see increased T cells, we didn't see increased B cells. It was mostly these macrophages that were giving us the changes in the immune cells. And then looking at what might be drawing these macrophages in, we also saw some extracellular matrix changes in the proteoglycans. So inter-alpha inhibitor is a proteoglycan modification that can be sort of stimulatory, and we found that modified proteoglycan was expressed in the parts of the valve that had these increased macrophages. So we thought so we know that fibrillin is an extracellular matrix protein, and so the idea is that maybe the matrix dysregulation is one of the things that's bringing the macrophages in that's driving the disease. So one of the lessons that we learned from the losartan story was that what you see in mice may not be what you see in pigs or humans as far as translating findings from mice into human patients. And so Andy Kim, who was the MD PhD student who was doing this work in the lab, he found a group in Japan that had a pig that had a gene-edited mutation that recapitulated Marfan syndrome in the pig. And so these pigs have the skeletal malformations that you would expect from Marfan syndrome, and they also have elastin dysregulation in the aorta, but they hadn't looked at the valves. And so Andy contacted them, and we started looking at the valves of these pigs, and what we found was that at 2 years and 4 years, these valves they're very thick, they have increased proteoglycan, and they're myxomatous, regurgitant, they have evidence of regurgitation. So this pig model does seem to recapitulate some of what we see in Marfan syndrome in human with regard to the valve disease. And so we see the matrix changes, and then what Andy also saw was that there are increased macrophages and monocytes, but not B cells and T cells in the diseased parts of the Marfan pig hearts, and they were increased relative to what we see in controls, although the controls have some. So that was actually as we expected. So part of the Marfan disease process in the pig heart valves also was these increased monocytes and macrophages. And then looking in humans, Andy also contacted a biobank in Canada that had some explanted human mitral valves, both from myxomatous disease of unknown origins, and also from individuals who had Marfan syndrome. And so in the Marfan syndrome disease valves, again, you see the mixing of the collagen and the proteoglycan, characteristic of myxomatous disease. And then looking at the immune cells, we see increased monocyte-derived macrophages in these human mitral valves as well. And so, again, that suggests that part of the disease in the human mitral valve disease also includes these monocytes and macrophages. So one of the things that we also checked was to see if the aorta had a similar increased infiltration of these cells. And interestingly, what we found was that although we could see the elastin dysregulation of both human and pig aortas, so the sort of the purple stripes is the elastin, and you can see that that's been sort of fragmented in the aortas of both the human and the pigs, we don't see CD45 cells. So if there were CD45 cells, you would see red cells in the middle panels, and there's no red cells. So what that says is that whatever is going on in the valves with regard to those immune cells is not the same thing that's driving the aortic disease. And so that was an important finding in terms of how you're going to manage patients with valve disease versus aortic aneurysm. So where do these cells come from? Andy did a bone marrow transplant study where to see if some of those cells were coming from the circulation or if they were just proliferation of the resident cells. And so here did a bone marrow transplant with tomato fluorescently labeled bone marrow and found that in the wild type, these cells do turn over. So you do see some of the cells coming from the bone marrow. And then in the fibrillin mutant valves, so the Marfan mutant valves, you see a lot of these red cells within the valve. And that says that during the disease, that these cells are probably coming from the bone marrow. And how do they get there? So one of the main mechanisms that bone marrow-derived monocytes target to specific organs is through the CCR2 receptor that's expressed on these circulating cells. And then the cells within the target organs are expressing the CCL2, the ligand for this receptor, which we also know is expressed in the Marfan valves. And so what we wanted to do was to look at mice that were mutant for the CCR2 receptor. And so that would prevent these bone marrow cells from targeting to these organs. And so then what we wanted to do is to see if the loss of these cells would then affect the progression of the disease. And so so these are the valves again, looking at the wild type and the mutant ones on the bottom, and so you can see these valves are thick. And then in the heterozygous situation, you can see that there are CCR2 cells that have gone from the bone marrow into the diseased valves. But in the knockout, so the knockouts are pretty normal in the wild types, which is another story, I think. But what Andy found a very striking loss of the sort of the thickening and the disease process of the fibrillin valves when this receptor was knocked out. And so what that says is that these cells do target to the Marfan syndrome valves, and they're required for the progression of the disease. And so now we think this is a potentially really interesting therapeutic target where these cells are it's possible to target these cells and then potentially prevent the progression of the valve disease. But we didn't see an improvement in the aortic, the structure of the aorta. And so again, supporting this idea that there's a difference between the valves and the aortic phenotype. So now we're thinking about in our model that this overall thickening dysfunction, but again, they're a target. We're interested in trying to figure out what some of these earlier events are, and that's something that we're working on. And so again, just to sort of put this in a clinical spin, we think the inflammation is starting sort of at the early stages of the disease. We see that in both the mice, the pigs, and the human patients. And then the idea is, could an anti-inflammatory therapy at this point stop the myxomatous valve disease progression? There are CCR2 inhibitors in clinical trials that we're now testing in the mice to see if we can inhibit this with a drug, which some of the pediatric cardiologists are very interested in, that may be a way to treat these human patients, babies now who have this severe myxomatous disease that they don't really have a treatment for right now. Okay. So I have another, hopefully short story, talking about calcific aortic valve disease. This is looking in the human. So again, now focusing on the aortic valve, where normally there's a tricuspid leaflet, it opens and closes, everybody's happy. And then with the diseased valve, often what happens is you get this calcification of the leaflets. The valve leaflets fail to open and close correctly. You have aortic stenosis. Right now, what you're going to have to do is get your valve replaced. And that's pretty much what we have to deal with these patients. Very high incidence, over 65 years of age, up to 10% of the people have, in the US, have severe aortic stenosis. As the population ages, this gets to be more and more of a problem, and we need new ways to deal with this. And what it looks like, a healthy valve normally is sort of translucent, has a lot of elasticity to it. This is a valve from a cadaver, and so you can see there are these horrible nodules, and basically this is, that's gonna cause you a lot of trouble. And as I mentioned, the standard of care is really replacement, or now more transcatheter interventions, where basically they just shove a valve construct into the context of this valve. And to me, that seems like a bad idea because, this calcification can break off, you can get a stroke, there's a lot of other problems with that. So it's kind of a one-shot deal. Once it's in there, it's not coming out and you're not getting another one. So that's, again, still work to be done to try and figure out ways to prevent this. So what are the things that are driving this? Again, the surgeons are like, we got this covered, but I think there's still a lot to do to try and figure out a way to prevent it or slow it or inhibit it, especially with people who are elderly who may not tolerate surgery very well. So how does this develop? This is in a human. This develops over the course of 60, 80 years, essentially. At this point, you have symptoms, but at the earlier stages, really, there's no symptoms. And so right now, the disease sort of presents itself here, and the surgeon's like, okay, we're gonna replace your valve, and now you're done. But the question is really, what are some of the things that are happening before the presentation of symptoms? And so we've been interested in this question for a long time. We're trying to figure out how to get at it because human valves are not, people with normal valves are not going to be giving up their valves to surgeons. So what are we going to do? So what we ended up doing was contacting a tissue bank of people. It's a biobank run by NIH that basically collects tissue post-mortem for people who have died from various causes. And so we were looking to obtain valves from the human valves from an intact heart. So the idea would be is you could get like the whole valve and look at the whole thing. And we compared that to surgical samples. So surgical valve replacement usually involves a surgeon going in and taking little pieces of the valves. If you're lucky, you get those pieces, and then you have no idea, like within the context of the valve, where it came from or what it looks like. And then you never get the piece that's like next to the aorta because the surgeons, they leave that there. So we compared what we were getting in those whole valves from the tissue bank to the pieces that we got from the surgeon, and then we also were working with a cardiologist who had sort of CT imaging images from individuals who are undergoing the TAVR procedure, so the catheter-based valve replacement. So we're trying to figure out, okay, can we kind of figure out what's going on early and relate that to what's going on later. That was the idea. So again, these are the samples that we got. So the post-mortem valves, no diagnosis of aortic stenosis, but we did get some individuals who had some type of cardiovascular disease and compared those to people who didn't have any reported incidents of cardiovascular disease. And our idea was the people with overall cardiovascular disease might have more valve problems than the controls. And then we compared that to the surgical valves. These are surgical specimens from valve replacement. So this was sort of the study design where from the biobank we got some valves from controls. So these are people who had no history of cardiovascular disease. The young controls tend to be like motorcycle accidents, young people, and then the older controls were from people who had cancer or some other kind of cause of death. And then we compared those two diseased valves. So our initial hypothesis, as I said, was these people who had hypertension or kidney failure or diabetes or bicuspid aortic valve might have more calcification than the controls. But what we found was that looking in the controls, so people who had died from various causes, anyone who was over 50, when we looked at the calcification, so this is alizarin red, what we're using as our indicator of calcification, basically anybody over 50, this is bad news for old people in the audience, there's calcification. Whereas under 50, so the people who died motorcycle accidents, gunshot wounds, whatever, they had less, they didn't have any calcification. So I think this calcification that we're seeing, so this calcification sort of at the hinge point, here's the aorta and this is the hinge and the distal tip. In the controls that were over 50, they had this hinge point calcification, also calcification out here. And then in the diseased, we also saw people with cardiovascular disease also had the calcification. But when we did the numbers, there wasn't a significant difference between the age controls and the disease. And we didn't see any difference between males and females either. So that suggested that our controls for calcification at least were not controls. So then what we did was we changed the way we categorized these valves, and we basically categorized them just based on how much calcification we saw. So if they had mild calcification, a little bit, but not a ton of it, or if they had a lot of it, we separated them into two different groups. All right. So what we did see was that there is an increase in age in sort of the mild calcification versus the more moderate calcification, and then we knew that the calcification really does increase with age. And again, bad news is that everybody over 50 is walking around with some kind of calcification. We don't know really how that kind of calcification relates to valve function. So that's something that is sort of an outstanding question. But, and then looking at this study was pretty underpowered, but is in terms of trying to figure out what possible comorbidities could fall out of it. The only thing that really fell out was age with calcification. So how does calcification progress? And so this is looking at, again, these sort of mild to moderate. And what we found, at least in this population, was that the calcification kind of starts at this hinge region and then extends out. And we saw a little bit of a leaflet preference for the non-coronary leaflet and the right coronary leaflet over the left, but essentially once you get to sort of this moderate stage, all of the leaflets are calcified. And then trying to figure out sort of histologically what the calcification looks like. So what we saw were potentially, what we're thinking, maybe two different kinds. So at the hinge point, we saw more of an intrinsic calcification within the sort of the confines of the valve. And then once you get out to the belly and the tip of the valve, you get these nodules. And so we're thinking that the nodules are really what sends you to the surgeon with stenotic valves. But a lot of people are walking around with this intrinsic, and again, we're thinking that may relate to like medial versus luminal calcification that you see with atherosclerosis, but we haven't really been able to nail that yet. And then looking to what the surgeons see with the CT imaging, these are patients who are getting ready to have TAVR, so they have aortic stenosis. And what we see with the calcification that you can see on the CT, I think is really mostly that nodular calcification that you see in the belly of the valve. So that's what the surgeons are seeing with the CT imaging. We don't really see what we're thinking of the hinge point calcification with this imaging. So we weren't able to see something that said, okay, these people are the people who have the hinge calcification. I think what they see with the CT and the resolution that they have are those really nasty nodules. And that's, these people are getting ready to have these valves replaced. And then trying to figure out how does this correlate with some extracellular matrix changes, looking at basically just how the valve leaflets are organized in terms of matrix, with the idea that maybe matrix changes precede some of these calcification changes. We didn't really see that. So even in the area that there are sort of these leaflet calcification, we do see the 3 layers. And within the area of sort of the leaflet calcification, we do see proteoglycan. So there is matrix within this intrinsic calcification. And then where the nodule is, you can see that's where the nodule is. But around it, there are some matrix changes, but we didn't actually see thickening of the actual leaflet itself. So the thickening is probably the leaflet plus the nodule, which is on there, and that's thicker. But the actual leaflet itself with the matrix didn't seem like it was that thickened. And then actually looking at the matrix, it seems like there is some differences. We see what we think are maybe like these cartilage-like cells in our imagination in this intrinsic region, which might be, the hypothesis is that that might be sort of an osteochondral-like bone formation. We don't have the data to really support that yet. And then in this nodular area, again, there are some matrix changes, but you don't see what we call these sort of chondro-like cells, these empty cells with the big nuclei in the nodular region. So again, there may be some differences in the cellular processes that give the intrinsic versus nodular, but we're not totally solid on that yet, and it's hard to get that from the human data, and the mouse models are probably not where we need to be able to tell that. But what we did see was elastic fragmentation, and again, that's sort of an indicator that things are going bad in the valve as the elastin gets fragmented. And so we did see that in the both the mild and the moderate cases, and then in the clinical there was really elastin problems. So there may be some crosstalk between the matrix and whatever's driving the calcification, but we haven't really figured that out yet. And then another thing that we saw was in the middle of the diseased valves were these adipocytes, and there were more of them in the diseased. It was an observation. Again, this is something that we really don't know what they're doing, but it does seem to be part of the valve degenerative process. And so another something to think about, I guess, that came out of these studies. The person who was doing this, we always looked at these diseased valves and like, what are the big empty cells? And so finally we figured out that they are adipocytes. They're not sort of neovessels. They seem to be fat cells. And so how they got there, what they're doing, we don't know. So just to summarize, this part of the talk, again, looking at these human valves, we saw valve calcification was a major thing in individuals who are over 50 years of age. There are sort of distinct ways that the calcification progresses. We think there may be 2 types of calcification, one pathologic, the other maybe not so much. And then we're looking at leaflet thickening and ECM abnormalities. But essentially, there's still a lot we don't know about what drives calcification, what are potential therapeutic targets. I think with different individuals, there may be different targets that are needed to be investigated. And again, what we're thinking is there may be different calcific processes depending on the individual. And that's something that we're still trying to figure out. So I'll stop there. The macrophage study was started by Alexei Hulud and then finished by Andy Kim, and Naju is working on the drug studies now. And Vicky Gomez-Stallins is the person who did the human study. I'll stop there.
Thank you, Dr. Yutzey, for this brilliant presentation. Always it's brilliant when you completely change your view of a field after hearing a talk. So, questions?
I start with thanks. The immune cell study is pretty interesting too. And so I'm wondering what happens. So you showed that most of them there in the disease seem to be coming from circulating from the bone marrow, I guess. What happens to the resident cells for one? And also, are these cells that are there during disease, are they showing a classical pro-inflammatory macrophage phenotype?
They do seem to be more, I don't know if M1, M2 is not in style, but they seem to be the bad ones. We haven't really looked at like the Lys6 profiles of them, but the resident ones don't seem, there is some proliferation of those, but the vast majority of those cells we think are coming from the bone marrow. So again, specifically what they're doing is something that we're trying to figure out. And again, doing the sorting and the single cell profiling is what we're headed towards on those that we just haven't gotten up with.
Thank you, Dr. Yutzey. Thank you, Dr. Yutzey. Amazing presentation. The receptor that
That's CCR2.
CCR2
Yeah.
that you suggested targeting, is there an existing small molecule or
Right.
antibody that has it been tested in humans?
So this RS3, I forget what the number is, but there is a small molecule that has been used, and the Boston group, so Swirsky and Narendorf, I think, are using it in. They've used it in atherosclerotic models, and also cardiac fibrosis and heart failure models, which these cells also seem to contribute to. And but clinically, it's in trials for cancer, angiogenesis. So it is in clinical trials. I think the safety trials have been done, and that's the molecule that we're testing now in the mice.
And what would the concern be with that in terms of non-specific? Like, is it a ubiquitous receptor, or is it specific?
Well, it would prevent targeting of those cells to any organ in your body that is essentially undergoing this response. So you figure it's probably there for a reason, but the side effect might be you cure after heart failure, and that wouldn't be so bad. So I don't know if the mice seem to tolerate it really well, and I think, like I said, the safety trials have been done in humans. So I don't know what the long-term effects of this would be. Talking to the pediatric cardiologists, they're really babies that are born with Marfan syndrome right now, they don't have anything that they can do for them that are that have severe myxomatous disease. And so the idea would be is that maybe you could put them on this drug for a little while until they get big enough and strong enough that you can do longer bowel replacement surgery. So
And the
that's
other
what we're
question,
hoping.
thank you so much. The other good question is osteopontin. Has it shown up in your RNA-seq? I saw periostin. Osteopontin is very much expressed in the bowel.
Yes, yeah. We didn't really see it as differential in the P7 to P30. But when we looked at the Clotho model, that was our number one hit. So in the Clotho model of valve calcification, they have vascular calcification, but they also have valve calcification. We did an RNA-seq a few years ago comparing the diseased valves and the normal valves, and osteopenia was the biggest hit in that screen. So it's doing something. We were able to shut down the calcification with BMP inhibitors in that situation, but the osteopenia is. Yes.
Can I scream? Do we wait until Sharon comes?
I'll run. I'm still under 50, so.
Yeah, I have to say, oops, is this working? I don't know. I don't know.
Well, now it's on, yeah.
Yeah, I looked around and I think I'm the only person above 50 here, so I'm really screwed. Yeah, in the audience, made me feel not so good. Josh asked my question, so I have to come up with a different question. I guess not necessarily directly from this talk, but all the models you've looked at, how it's a big question. How do you think that these interstitial cells, how plastic are they?
Yeah, so that's a question. And like I said, we're getting ready to do single cell on the Marfan models. And then there's still this question that we've been talking about for many years, like in these myxomatous models, it looks like there's maybe more of these melanocytes. But it could also be that the macrophages ate the melanocytes and now they have more melanin in them, which may be why you get those sort of, I didn't show that today, but we've seen that in the past where I think there may be some interaction between the melanocytes and the macrophages at the tips of the valves, which is why a lot of people in these disease models see more of what look like melanocytes, but it looks like maybe the macrophages ate the melanocytes, and maybe that's why they have melanin in them. But we're still trying to figure that out.
During normal development and during normal patterning, do you think there is a need for these cells to be plastic?
I would expect there would be, and we know that when we put valve interstitial cells in a dish, we can get them to do all kinds of things depending on what we stretch them or add stuff or whatever. So I think there is plasticity at some level, and then the question is, if you fate map, the collagen interstitial cell, does it turn into the glycosamine glyco-interstitial cell? And we haven't figured that out yet. We have some ways we might be able to do that with we have a periostin inducible Cree, and so we could look at that and see if it converts to another kind of cell. We just haven't done it yet.
I was going to ask that question. So, but also in the disease.
See, we think alike.
Yeah, good.
I was wondering if you get one to turn into the other. But in the disease, though, have you looked at any of the things that you found from your single cell analyses in the mouse during development? Have you looked to see if you see any of those markers in the human disease valves that may suggest that some of these phenotypes you see there are related to those?
Yeah, and we've spent a lot of time trying to get that to work. These diseased valves that are formalin fixed, it's hard to get the antibodies to work to the level where you could really convince yourself of anything. And we probably spent 6 years trying to do that. So we're still trying, but yeah, the antibody studies on the disease valves have been really hard. So we've done those experiments. Whether it's convincing, I'm not convinced. So, here's one back.
I'm good. So a simple question where I guess everyone can follow a little bit better. How come mouse valve leaflet sections seem so thin towards the base and really thick towards the free edge, whereas human sections are pretty the same thickness throughout? Is that just an artifact of the way you section these, or is that an actual thing of mouse valve leaflets?
I think it depends on where you are in the tissue. So when we did the whole valve leaflet from the human, you can still see sort of a thick hinge, a thin belly, and then a thick distal tip, which I think it goes with the cup shape part of the valve. And then we see that in the mouse as well, but the mouse valves are so small. I mean, that belly region is maybe, I don't know, 3 or 4 cells thick, whereas in a human, you just have a lot more real estate because it's bigger. So I think overall, the shape is similar, but not exactly the same. And then when you think about it, a mouse valve, I mean, that heart's beating 400 beats per minute, so that valve is just, it's just going, right? And then a human valve at 80 or whatever is a lot more. It's slower, and then it also, I think it undergoes a lot more of the back pressure, maybe more so than the mouse valve. But this is a biologist talking to an engineer, so again, I think that the pressure that the valves are under does determine their structure at some level. And interestingly, like if you do the Ross procedure and you put a pulmonic valve into an aortic position, it gets fatter in a human. So again, there is some plasticity in the structure. But yeah, you're right. The mouse valves and the human valves are not the same. That's just the way it is.
So I have a question regarding the immune cells. So you showed very nicely that the leukocytes, they don't contribute towards the ECM formation.
Mm-hmm.
But I guess, is there any evidence to suggest that there may be perhaps an indirect role as far as perhaps the immune cells, working with the interstitial cells in a sort of a paracrine regulatory manner to dictate whether or not the ECM would be normal versus pathological?
Yeah. And that's something that Nakano's group has looked at a little bit, but also in those CCR2 knockout valves, in the wild type situation, it looked like there might be a change in the collagen. We haven't looked at that in real detail, and that would be the situation where if those circulating cells are missing, is the valve structure affected? And maybe subtly, I think that's possible. And then the resident guys, we haven't ablated those. So we were trying to do that, didn't work, anyway. So we're trying to ablate cells and actually, but for another project, we're like killing periostin positive cells with a fibroblast, but we should also be able to kill valve interstitial cells. That wouldn't answer your question about the immune cells, but maybe some crosstalk mechanisms and maybe a regeneration mechanism. So we might get that as a side effect of some of the other studies that we're cardiac.
Any other questions? If not, I'd like to thank our speaker once again. Parting gift for you. Thank you so much.
Thank you so much. All right. Thank you.
And before you all leave, I was asked to announce there's a couple of important people outside, with tables. Please do visit them. There's also a blood donation happening. So Heart Day is a cardiovascular event, both figuratively and literally. So if you want to donate blood, the truck's outside. Thank you.
I want to see the gift.
There's a 3D printed heart.
Oh, wow.